Collect better clicker questions. Maybe, maybe not. You have just discovered a new protein. It has 25% amino acid sequence identity to another well-characterized protein. This means that the proteins definitely have the same structure. The proteins definitely have different structures. They're all alpha helical. Both proteins are dimers. None of the above. And we will start. Yes? Feel free to talk amongst yourselves. This is supposed to be a group thing. So you have another minute. Talk.
According to your TA,
10. 5. Okay, so what do we think? Whoa. So, unfortunately, I need to get this set to be a little bit more transparent. Proteins definitely have the same structure. Very few people seem to think that. Proteins definitely have different structures. We've got quite a few people who think that. And then they're all alpha helical, the proteins are dimers, or none of the above. None of the above is always a good guess if you have absolutely no clue for one of my questions. And in fact, in this case, it is correct, because you can have proteins that are 25% identical that have identical structures, and you can have some that are 25% identical that have completely different structures. So you don't know. There's no way to tell which ones of those are correct. So E is, in fact, the correct answer in this case.
Even if there were 30% or more?
30% or more is that's where the cutoff is. So that the comment down here was 30% or more. If it's more than 30%, they're going to have pretty darn similar structures.
Right.
Yeah. So if I said 40% identical or 50% identical, say at some point in a couple of weeks, then I would say it definitely has the same structure. 30% is really kind of borderline, so that's why I had 25% here. So our next clicker question. The transforming material in Griffith's experiment transformed mice, phage, bacteria, E. coli, or Sulfolobus, my favorite organism. Again, feel free to talk about it. I heard people down front saying you could look it up in the book. Sure, you can, but you won't be able to do that on an exam. You also won't be able to talk to your neighbors on an exam either. So. It's totally specific. There is one and one correct answer in that.
Isn't it?
There's no answer for the DNA up there.
No, what did it do, that transforming material, which we don't know is the DNA, but what did it transform?
That's what I mean. It's not specific enough. The phage is how it
Hershey-Chase was phage.
Okay.
That's right.
So, most of you agree with me. Yes, it is the bacteria which were transformed. In fact, the figure in their paper, which actually has nothing to do with the mice whatsoever, is your little colonies to the big smeary colonies. Turns out that gives you the virulence, which is what you see in the mice in Griffith's experiment. Phage is Hershey-Chase, the meeting I was at all last weekend. E. coli, of course, is a bacteria. It's not the bacteria which is causing disease in this particular case, and I have to have something for E, so sulfolobus it is. You can transform sulfolobus, but not with this particular experiment. We, in fact, transform sulfolobus all the time. And in fact, quick advertisement for the recombinant DNA techniques course, the lab course, which we're doing next term down at the CLSB, you will be transforming sulfolobus with transforming material. So there's a question over here.
Yeah, I just had a question. I was looking at the syllabus, and what's your policy on clickers? Because apparently now we can talk. I just don't know.
I sorry, should have said that right at the very beginning. You're more than welcome to talk with clickers.
Okay.
Yeah.
So is it like a maximum amount of points, or how does the grading work?
Oh, it works just like the exams. So high score, and each of you has an individual clicker, and each of you has an individual point score. And so I just take the high point score that's set as 100. Everything scales from that.
Oh, okay.
Yeah.
So I answered bacteria, but
Yes.
I think the question maybe taken another way could be the phage was sort of doing the transferring of DNA from one bacteria to another.
Avery, there was absolutely nothing to do with phage. Avery and Griffith had nothing to do with phage. Phage only was Hershey and Chase later. So sorry if Eric didn't do a good job of explaining that. I can blame him for that now, and you can blame him too. And yes, I made up the clicker questions, not him. So I can't blame him for that. You can blame me for those. Okay. Let's move on and talk about Watson and Crick. Particularly important as far as Watson and Crick's model, and it was just a model. They didn't have direct data. In fact, they may have even stolen the data, which they used to come up with their model. But the real key here, of course, is base pairs. And in the past, I've asked people to draw base pairs on the first exam. There are too many of you to draw too many base pairs. I'm not going to do that. But I might draw an incorrect base pair and have you tell me what's incorrect about it. So I do expect you to know that. This will be, I know it sounds like a really, well, I won't use the S word, but not necessarily a really good idea that you have to memorize these kinds of things. But it turns out that the base pair, and we'll talk about why that is as we move on through today's lecture and future lectures, it's really important that first concept, but also the absolute chemical structure of that, because it really determines how DNA and protein interactions with each other, RNA, DNA interactions, et cetera. So the structure of the base pair is really fundamental to understanding molecular biology. Part of that has to do with denaturation and how you get the two strands to come apart from each other, which is critical for karyotypes that we'll talk about a little bit later on. We talk about chromatin, and then today we'll probably end up finishing up with nucleosomes, but if we don't, we'll just keep talking about that on Wednesday. So what did Watson and Crick know? They knew the structures of the nucleotides, which we'll look at on the next slide. They knew that they were linked together with 5' to 3' phosphodiester bonds, and that gets us back to our molecular biology polarity, a front end and a back end. And so 5' to 3'. We're going to be talking about 5' and 3' all the time. And then Edwin Chargaff's rule, which actually he never called it a rule. It was other people who said that. And that was that the concentration of adenines was really similar to the concentration of thymines in DNA. And the same thing was true with guanines and cytosines. Probably their real insight here was that they were able to put the two of them together and really to figure out what was going on there. what did they have? They had fiber X-ray diffraction of DNA. This was Rosalind Franklin's work. A lot of people discuss, why didn't Rosalind Franklin get the Nobel Prize for this? Unfortunately, she had died, probably due to the radiation which she'd exposed herself to in gathering some of this data. And there's always been discussion. There's a book which I didn't bring with me. I'll bring it on Wednesday. The double helix, Hunt for the Structure of DNA, where Jim Watson sort of denigrates the contribution that Rosalind Franklin made. Later on, in fact, in their careers, they worked really closely together and had great collaboration as far as that was going on. So she and her student, who published their paper back-to-back with the model for DNA in Nature, had these two structures, actually. Fortunately, the projector doesn't do a very good job here. But up at the top here is one of their X-ray fiber diffraction pictures, and down here at the bottom is another one, sort of the classic one, but this is probably the one up here, which Maurice Wilkins, who was sort of Rosalind Franklin's boss and kind of collected all of the laurels. Everyone hears about Watson and Crick. How many people hear about Watkins? Wilkins, sorry. See, I even mispronounced his name. He ended up getting the Nobel Prize as well for this. But basically, from these fiber diffraction images, and if you're interested, we can go into exactly why this is. If you want to know even more about it, take some of the crystallography courses in physics. But basically, it's a 2 nanometer diameter filament with a helical construction. It's that X which tells it it's helical. 2 nanometers is basically this big reflection that you have out here at the end. And then the 3.4 nanometers are the spacing between each of these bands that they have here. Linus Pauling, who not surprisingly was really into helices because he'd come up with the alpha helix a number of years before, also had a proposed structure for the structure of DNA, but it was flipped around and it had 3 strands rather than 2. So that's what they had. There are a couple of other papers as well, but that was basically it. Let's talk a little bit more about this 5' to 3' business. This is all about the structure of the sugar. So the 2' deoxyribose, again, it's DNA, so it's the deoxy. That's this position right here, the 2' position. Prime here just stands for the position on the sugar. If you talk about the 1 position, 2 position, 3 position, 4 position, that's having to do with the base, which is up here. So again, as far as the backbone, the polarity is concerned, you've got a 5' end and a 3' end. It's true for a polynucleotide. It's also true for the individual monomers that you're putting together in the process. In DNA, you've got 4 different bases: adenine, guanine, cytosine, and thymine. Although some of you may have read, the synthetic biologists have come up with 2 more, which is really kind of cool. Again, we'll talk more about that later online if people are interested. And we knew, and again, from these were from Chargaff's experiments, if you just purified all of the individual nucleotides, then you had always the same amount of 1 purine and 1 pyrimidine. I always remember these. Purine, short name, big molecule. Pyrimidine, long name, small molecule. And of course, they can only fit together if you've got 1 pyrimidine and 1 purine. As a quick aside here, this is an example of one of the panels that's in the textbook, which I think does a really nice job of explaining all of the individual nucleotides, the bases, and the sugar here, just basically for review purposes. This is what one of those strands look like over here. And again, 5' here, 3' over here. So they're always attached in this orientation. And the orientation is critical. If you didn't have an orientation, you didn't have polarity, you couldn't use this as genetic material. That information wouldn't make any sense. It has to be a linear information stream. And so that process, always 5' to 3', allows you to be able to use DNA as a genetic material. So what did they do? They built models. And actually, again, if you read their book, they got the machine shop at Cambridge to build their models, and then they put them together. And we'll see the actual model that they made a little bit later on. And then they came up with this idea and then they got Crick's wife to draw this picture. And so this is, in fact, her is unfortunately not cited in the paper. But here, two complementary strands, so it's a double-stranded helix as opposed to the three-stranded one that Linus Pauling had. And they're wrapping around each other in anti-parallel. And so anti-parallel is getting back to that 5' to 3'. So one end's 5', the other end there is 3'. And so I always do this with my two arms, back to front. 5' over here, 5' over here. And so anti-parallel, and that was really critical. And that's something that you can't get from that fiber diffraction. You have no idea that they're anti-parallel relative to each other. That's what you get by model building. That's how you get these by putting these things together. And the phosphates on the outside, bases on the inside, and the main thing there is that again was opposite from what Pauling's structure was. And then the hydrogen bonds in the middle, which are perpendicular to the middle of the double helix. Turns out that perpendicular is wrong. If you actually look at the structures, they're not completely perpendicular, but that's what worked out for the model. A couple of things they had to do, and again, this had to do with making the model nucleotides. And again, we'll see those in just a second. And it was where each of those bonds could actually form. There are tautomers that happen in all of these bases, so C4 and C6. And again, this is not prime. This is the absolute numbering in the bases. Are in amino groups and keto groups in guanine and thymine instead of enols. We'll look at the major and the minor grooves. You can see them on here actually really quite nicely. The major groove is this big gap right here, where you've got this arrow going upwards, and the minor groove is between the two strands here. And what this says is also it's an asymmetrical structure. And in fact, you can fit a third strand down through here, but it doesn't do nice base pairing in the process. So I say not recommend, I require that you read the paper. I'm not quite sure how I come up with questions from the paper, but you'll see some of the ones that I've done if you look at some of my old exams. Also wanted to mention the handedness of helices. We mentioned, or I may have forgotten to mention, or maybe Eric mentioned this, is that all alpha helices are left-handed helices, but DNA are right-handed helices. And there are lots of different ways of thinking about left and right-handed helices. A few years ago, I asked people to bring in examples of left-handed helices, and they found these things, and who knows what this is? Are you old enough to know what one of these is? So this is, in fact, a left-handed helical telephone cord. Most of them are right-handed. Next time you go and check, you can take a look at them. It's also a great model for looking at supercoiling. So there are a couple of ways of thinking about this. One way is if you look down the helix and you see which direction it's going, clockwise or counterclockwise. So in this particular left hand, it's going counterclockwise, whereas the right-handed helix would be going in the clockwise direction. And the thumb process, which is what they have down here at the bottom, where if you look at how it's wrapping around, I'll leave this up in front, you can come take a look at it a little bit afterwards. If you look at a right-handed helix, the way you wrap the fingers around, if they're going in the direction of the thumb, that's going to give you whatever hand that happens to be. So in the case of your right-handed helix, be wrapping up that way, and the left-handed helix, it's wrapping around in the other direction.
Does right-handed mean counterclockwise then?
But it depends on which way you're looking at it, whether it's counterclockwise or clockwise. So again, that's why I always say when you're looking down the helix and it's going away from you in a counterclockwise direction, that's left. Okay. And I'll also bring, we've got some models of DNA that I'll bring, and you can take a look at those as well a little bit later on. And this is also just the top as a reminder for those that general chemistry, what your ketos and your enos are. So again, in terms of the base pairing, and the only way that this works in terms of base pairing is if you have the amino groups and the keto groups for things like cytosine and guanine. And that's because of the base pairing which happens there. The original model looks like here on the right-hand side. It's literally about 3 to 4 feet tall. They have a copy in the Smithsonian. I don't know if anyone's seen it. Anybody seen the replica of Watson and Crick's model? It's pretty big. But anyone recognize this reference over here at the bottom? Yes? It's only a model. And that's, again, I misspent my youth. But it is important to remember that it's a model. It's not what DNA looks like, and it's in fact not what most DNA looks like most of the time. It's a great description for understanding sort of the basis of how DNA strands fit together. Gives you some great insight in terms of how replication takes place, but it's only a model. And so there would need to be lots more data to come up with getting a more precise and still even a model of exactly what DNA looks like. So again, base pairs, unfortunately, our textbook and most textbooks don't have a nice model that has absolutely everything on them. I like to see all the hydrogen atoms, et cetera. But again, the main thing here is that we've got 3 base pairs that form between guanine and cytosine, and 2 that are forming between adenine and thymine. And of course, with this, it doesn't matter which way around it is. So either way, you end up with a structure which is basically identical, determined by where these sugars are, because the sugars are attached to the phosphates, which are the backbone. So this position of this sugar and that position of that sugar are identical to each other, whether you have a GC base pair or an AT base pair, or if you've got a CG base pair or a TA base pair. So they're always going to be at exactly the same place, and that means that the DNA can have the same structure even though the actual chemical information, the bases in the middle, can be different relative to each other. And so that's the real beauty of this particular structure, is it gives you that symmetry. And this is a quick aside. Originally, Watson and Crick actually thought that there was a base pair, or say a hydrogen bond over here, and it turns out they were wrong. But one of the ways that we're wrong. And again, hopefully I'm not beating a dead horse on this. This only works if you've got anti-parallel strands relative to each other. So 5' end, as we have here in this example, up at the top here, has to base pair with a 3' end over here. And again, otherwise that doesn't work in such a way that you have the sugars in exactly the same positions on either side and being symmetrical. So you're always going to have the 2 strands opposite to each other. When people have drawn base pairs in the past, very often they end up with them parallel to each other. Not good. They always have to be anti-parallel. And again, just one more example of this. Here also, 5' end versus the 3' end. And I wanted to emphasize here the major versus minor grooves. This is now in the space-filling form or the van der Waals interaction form. Another thing to see here is that there's actually not much space between this. It's very tightly packed in the middle of the double helix. And it turns out that that packing actually probably helps the strands to stay together at least as much as the base pairing does. The base pairing probably has much more to do with the specificity of what base is where, as opposed to the real stability. Everyone talks about how do you get the DNA to be stable. Well, actually, it's a lot to do with packing forces. It's that little extra stability you get from those small interactions. And so again, like we talked about before in protein structures, lots of small, weak interactions, hydrogen bonds, hydrophobic packing, et cetera, which are holding these things together. Yes?
So when you're looking at this picture,
Yeah. Okay, so the problem with this is that it's in 2 dimensions. Okay, sorry, back up, so this is actually on recorded. Are there multiple major and minor grooves? Again, it's just sort of a way of paraphrasing your question. No, there's only 1 minor and 1 major groove. It just wraps around the helix, and that's why having a 3-dimensional image would be much better here. So I'll just sort of try and trace it here. So if you look at the minor groove here, it's this thing here, and that wraps around here. It goes in the front, and then it's going to go behind and comes out down here again. So that's your minor groove. Yeah, so but there's always going to be one which is smaller and one which is larger. And so, and again, that's why I need to have a model to take a look at this. And that does make a big difference in terms of that. And we'll see why major and minor are important in just a second. But again, the major groove here, so that's the major groove here. It's the same major groove here. It's the same major groove here. And so it sort of wraps around your helix. It's part of that process. So it's actually even a little bit easier to see in this image, which is from one of the other textbooks. But again, you've got major, minor grooves. Major one is right here, and then you'd wrap around, it's going to be the back side. It's going to end up being right here as well. And then the minor groove, which is right here in between these guys. So why am I beating on this? Sorry. Okay, so why do you have a major and a minor groove? That has to do with, again, it's the model building process, and the only way you can get them to fit together in such a way that, A, they're stacking on top of each other. So it's a nice example here in this image that these van der Waals interactions are really stacked right on top of each other. If you have a completely symmetrical helix, which is one of the models that I didn't bring, because if you line everything up right in the middle, then you end up with completely symmetry, and so you don't have a smaller and a larger one. But it has to do with where you're binding to the sugars. So actually, I should have it over here. So each of the sugars from each of the two chains, they're only going to be in the same position if it's slightly offset. So if you look at where the bases are, you'll notice they're not completely in the middle here. They're slightly offset. So over here, they're a bit more on the right-hand side. Here, a little bit more over on the left-hand side. So they're not completely in the middle. And that's what gives you the major and the minor grooves. So this just has to do with the chemistry. That's how you can fit these things that fit together. And it turns out to be incredibly useful, and that's probably why nature in 4 billion years has selected it to be that way. The other thing, and this is getting a little ahead of myself here, I mentioned that major groove, minor groove, it's a bit larger in the major groove. Turns out it's exactly the right size to put an alpha helix into. Is that really surprising? Again, probably from an evolutionary point of view, it makes a lot of sense that proteins are going to be interacting there in the major groove of DNA. By no way is this the only kind of DNA which you find. There's also so-called A-form DNA, which you find at slightly different salt concentrations. It turns out that the best pictures that Rosalind Franklin had were, in fact, of A-form DNA, which is over here on this side. Again, two-stranded, right-handed helix, but the bases are not perpendicular in the middle here. They're really skewed relative to each other. It's got a much narrower but deeper major groove here. And it turns out at different salt concentrations, you can have DNA with these very different structures. But for the most part, again, this model is close to what double-stranded DNA looks like most of the time, and usually you've got a pretty wide major groove. So what's in this major groove? This is really, again, probably getting back to your question about the majors and the minor grooves. Why have a major groove and a minor groove? Well, basically because you can get all the information about what a base pair is just by looking in the major groove. And that looking in the major groove usually, again, has to do with an alpha helix, or sometimes it's 2 beta strands, which have then amino acids that can then bind to or interact with those bases which happen to be there. And so here is just an example of that, looking at a number of different base pairs. And basically the idea here is that if you look at the arrangement of atoms and what kind of bonds you can make in the major groove, it's different for all of the 4 different bases. So here, hydrogen, hydrogen, positive group, negative, negative. Again, these are your oxygens. Here, other way around, again, just looking in the major groove, you've got a hydrogen over here, a couple of oxygens, etc. Same thing is true with A's and T's. Now you've got a methyl group that's sticking out in the major groove. If it's on, in this case, the left-hand side versus the right-hand side, that tells you what it is. So you don't actually have to take the bases apart to get the information that's there. The base pairs hold them there, but if you just look in the major groove, it tells you what's there. However, in the minor groove, you can't tell what those bases are. So if you want a specific interaction with double-stranded DNA, it has to be in the major groove, because that's where the specificity is, not just of finding the base pairs, but also any kind of interaction that you want to have. That's just shown here in a cartoon form. Here, if you look at a GC base pair, you've got the keys down here at the bottom, hydrogen bond acceptor, hydrogen bond donor, etc. I don't expect you to be remembering exactly this code, but if you know your base pairs, then you'll know what these things look like. Any questions on sort of major groove, minor groove DNA structure? Because otherwise we're going to move on and talk about some more things here. Yes? Okay, so the question is, do A and B forms of DNA have different functions or just about the same? I'm going to expand your question a little bit and tell you about zDNA that I don't have a picture of here, but it turns out zDNA is a left-handed helix that forms in DNA. And people argue a lot about whether that's actually something which is physiologically relevant. But it certainly gives you some very different kind of chemical interactions. And so even if you think about A-form DNA, those hydrogen bond donors and acceptors or methyl groups are going to be in very different places relative to each other. And so if you've got a particular protein that's got side chains that are in a particular place, then it may well bind much better to A DNA as opposed to B DNA. And that's, again, that's getting back to the whole, the B DNA as a model. It's wonderful. But that's not what DNA looks like. If in, well, getting a little ahead of myself here, but if you think about the human genome, if all of our DNA was B-form DNA, the genome in any given cell would be about that long from the floor to here. So that can't be true. So again, that's a sort of, a lead on into some of the other stuff we're going to be talking about there. Other questions about the DNA structure, base pairs, et cetera, before we move on? Okay, so this is something that's one of the fundamental properties of DNA. Mostly has to do with the base pairs. They're in the middle. If you just look at the absorbance that you have in the UV, which is mostly due to bond interactions, all the double bonds that you have in the base structures relative to each other and how they're stacked on top of each other. If you have double-stranded DNA, that has a particular absorbance, but it turns out as you pull those two strands apart and have single-stranded DNA, now these bases are not as nicely stacked on top of each other, so it ends up actually having a higher absorbance in the UV than double-stranded DNA does. And again, that has to do with all of the electrons interacting with each other. What this basically means, and this is important in terms of looking at DNA, how we do experiments with this, is that if you have double-stranded DNA, it's got a relatively low absorbance. But if you have single-stranded DNA, it has a much higher absorbance. And what you can use this for is actually really cool, is you can now heat up your double-stranded DNA. And at a certain point, the absorbance of your solution is going to get much higher. And it turns out it does this very rapidly over a relatively short time window. And you can just get the midpoint of this transition. It's called the melting temperature, or the TM. And again, it's a very fast, both kinetically but also in terms of relatively short ranges of temperature, where you get this melting that actually takes place. So the melting temperature, again, this is the midpoint of that. One of the couple of really neat things about the TM is that this is completely reversible. So you can heat up your DNA. You heat up your DNA, your absorbance goes up. You cool down your DNA, the absorbance goes down, which tells you you're getting double strands that form. You heat it up, goes up, and so on and so forth. So it's a completely reversible kind of thing, which is really kind of truly amazing if you think about how complex a lot of these DNAs are, and they're coming together in the right way. So your base pairs are actually all forming appropriately. But it's also really useful if you now just plot the TM, so that middle transition, I'll get back to you in just a sec. You can also correlate this with the percent GC that you have in the DNA that you're looking at. So it basically also tells you that that transition point between double-stranded and single-stranded really depends on those hydrogen bonds that you have there. So if you've got more base pairs that have 3, you're going to be pushing that TM a little bit higher. Yeah, your question. So the question is, can you do this with proteins? I guess is just to paraphrase your question. And the answer to that is, for some, yes, but for a lot, no. I always sort of use the example as it's really hard to unboil an egg. But for DNA, it turns out that it actually is really reproducible. You can pull them apart, put it back together, pull it apart, put it back together, pull it apart, put it back together. And it turns out that's really critical for replication, because you're going to have to be able to separate those things and get them to come back together as well. And it turns out it's extremely useful. We'll talk about karyotypes here, getting down to the questions in the front row that we had before class. And also exactly really, really useful in terms of tools for trying to get an idea what piece of DNA are interacting with each other. So again, you can get GC. It turns out that the salt concentration also is going to determine what conditions you get these two coming apart, and that salt and pH to some extent, but it's pretty pH independent actually. But the salt concentrations is also going to vary a little bit depending on exactly where you are in the cell, but much more importantly in your test tube. And so you can change also the temperature that you get this transition that's taking place at. What that tells you is there's actually a lot of salt molecules that are interacting with the DNA. Now, the outside are the phosphates. What's this phosphates charge? Negative. So what's it going to be associating with? It's going any kind of positive ions which are around, which are usually sodium and potassium, particularly potassium inside the cell. And so it turns out that those salts, and changing those salts, will actually stabilize or destabilize your double-stranded DNA. Yeah? So at different salt concentrations, you're going to be changing the TM. And we'll look at that just in the next slide in just a second. Pardon? So we should, the comment down here is just say you should eat more salt. For the DNA point of view, if you want to really stabilize it, that might be a good idea, but there are all kinds of other things that salt's not terribly good for. So ask your physiology instructors about eating too much salt. But again, it's probably much more useful from the point of view of the molecular biologist in the lab, ignoring all the messy cell stuff. Then more salt's usually a very good thing. Was there a question in the back? Or just a stretch? Okay. Okay, so again, now this is sort of more of this process. How do you change the TM? What can that kind of experiment tell you about what DNA you have? Turns out that if you have DNA that doesn't pair well at the ends, that will really destabilize your DNA. But if it's not pairing well, again, you don't have G's and C's in the middle, it pairs extremely poorly. And in fact, that will lower your TM really, really drastically. And just by putting DNAs together and seeing how fast or how slowly they come apart can tell you a lot about these DNAs without even really knowing anything about the sequence or anything else like that. And again, this is the salt issue. If you just look at low salt versus high salt, lower salt conditions, it's easier to pull your DNA apart. And at high salt concentrations, it's harder to pull your DNA apart. Although if you go to too much salt, that actually is going to push those strands back together again. So this is, so you don't want to be eating this much salt, getting back to your question. But over here, that might be stabilizing your DNA a bit. But if you want to be getting the DNA part to replicate, it actually might be a bad thing. So, but again, this is much more important for the molecular biology aspects of things, is being careful about the salt concentrations as well. So let's talk a little bit about how you go from this DNA, which is longer than you, if it really was all Watson and Crick form, how you can actually fit all of that DNA into cells, and in eukaryotic cells, actually nuclei, so it's smaller than cells. It's all about the chromosomes. First, we'll look at some of the chromosomes. We'll look at some karyotypes. We'll be looking at these chromosomes. Talk a little bit about chromosome evolution, but mostly about how you can go from B-form DNA to really compact forms of DNA. So what do you call this? It's called chromatin, and chromatin is just the DNA plus how you compact it, and how you compact it is almost always proteins. And this is why, hopefully, Eric talked about this on Friday. People originally thought that protein was the genetic material, because if you look at chromosomes, people knew that chromosomes were the genetic material, and all the cool stuff, at least as far as they were concerned, in the chromosomes was these proteins. So it's the proteins which are really allowing this compaction. So you have to compact it, but it's really kind of a complicated problem because not only do you need to compact this DNA and get it to a much smaller form, be able to fit into cells, but at the same time, you have to have access to it by all of your replication machinery, getting to that major groove that I was talking about, how you can get an alpha helix to come and fit in there. So it's actually a really complicated problem, and we still don't completely understand, actually, how you can get compaction and still have access. We'll talk about what we do understand in the next couple of lectures, but it's still a really open question. At the same time, you need to protect DNA. When we talk about DNA repair, DNA is actually incredibly susceptible to all kinds of things that we happen to live in. The major one of those, of course, is water. And DNA is incredibly susceptible to hydrolysis. So chromatin actually helps with that. Also helps to organize where the DNA happens to be inside a cell. And then finally allowing you to separate those two strands relative to each other. Bacteria have exactly the same problem that eukaryotes do. Again, they've got to compact their DNA. They have to make sure it's accessible to all kinds of other proteins. People talk about chromatin. They almost always think eukaryotic chromosomes. It's not the only thing at all. Certainly bacteria. And it turns out the more we're learning about bacteria, we are finding that a lot of them do have compacted structures. People call them nucleoids. They just don't have a membrane around the outside, so you don't call them a nucleus. Yeah, Travis. Do viruses have chromatin? Always. Take my virology course next term. I know you will. But yeah, so as soon as we're talking about compacting DNA, or in a lot of the viruses, they have RNA, it's going to be that nucleic acid protein complex. And so you can talk about viral chromatin. There's one particular one that turns out uses nucleosomes too. Totally cool virus, but that's getting ahead of ourselves. Yeah, and okay, the other question, do archaea? Yes. Okay, so I would say the nucleus, so a lot of people think about bacteria as being basically sort of a bag that's everything is mixed up in it, all coherently distributed. Turns out that's because our microscopes were no good. And now we've got higher resolution microscopy. In fact, the Nobel Prize this last year for medicine was the higher resolution microscopy, super resolution microscopy. And part of that was figuring out that there are, in fact, subcellular compartments in bacteria. They're just not as obvious as you see them in eukaryotic cells. And they're also not as membrane bound, or at least not very many of them are membrane bound. Okay, what are this compaction? Oh, this is not great projection here. Sorry about this. But if you look at now eukaryotic cells, because we actually know a lot more about how these things work in eukaryotes than we do in bacteria, the compaction here, basically, this is your almost naked DNA that you have down here at the bottom. This has got nucleosomes that are associated with it. Most of the DNA in an interphase nucleus is going to be in this sort of 30 nanometer fiber form. And then it's just by looking at cells. Looking at cells in the electron microscope, this is now an interphase chromosome. Most of your DNA is going to be in these 30 nanometer fibers. And of course, in metaphase, you actually can see these now in a light microscope. That's the absolute compaction. So it's about 100,000 times compaction that you get between nice, happy B-form DNA. And one of the things that I forgot to mention there, but it's a good way to lead back into it, is if you look at Rosalind Franklin's beautiful fiber EM. You remember there was a 3.4 angstroms and a 34 angstrom repeat? Those, as it turns out, are the distances between each of the individual bases. That's your 3.4 angstroms. 34 angstroms is every 10 bases. You go round once, and you basically repeat. So if you have 10 bases per 34 angstroms, that's how I get to my 2 meters for 3 billion base pairs. So it's that compaction where you've got to go from 10 base pairs per 34 angstroms down to a thousandth of that. And that's what, again, most of your genome is most of the time. or 10,000 when you need to have metaphase chromosomes and separate them from each other. So that brings us to what's on these individual chromosomes. And the way that you can look at that is, fortunately, because when you do again that 10,000-fold compaction, you can actually visualize these under the light microscope. And so that gets us to karyotypes. This is, in fact, from the 4th edition of the text, but yours looks basically identical to this. This is a human set of chromosomes, and just a haploid set because it's one of each. 1 through 22. These are which all of us have. A few of us have this little extra guy hanging out over here. The more superior people have 2 copies of this one. So autosomes are those, again, that everybody has. There are always pairs of them, the male and female, and then the so-called sex chromosomes at the end. This is what's true for humans and most mammals. Turns out that in different organisms, you've got all kinds of different sets of numbers of chromosomes. The numbers of chromosomes varies incredibly wildly. But of course, not surprisingly, we're anthropocentric, so we're interested in human chromosomes. Yeah, right in the back. Yes. So the question is, again, just for posterity's sake here, don't other animals have different, they're not always X and Y? There's a huge variety. We're going to talk a little bit about sex determination in fruit flies, which are XXYY. But you've also got Ws. You've got all kinds of different kinds of sex determination. It's a really fascinating question. In fact, Dr. Estes here in the biology department is interested in how sex sort of arose, because there are also some organisms that reproduce sexually, some that reproduce asexually. And evolutionarily, it's a really fascinating question, how that actually could have and actually has clearly happened in some cases. So this is really beautiful, that we've got all of these chromosomes all laying out, next to each other here with this nice banding pattern. This is what people actually found more or less at random. Turns out that it has something that stains actually really relatively well. What we found more recently, and this is by using that case that you can take the 2 strands apart and put them back together, you can get individual small pieces that where any given sequence is in any given chromosome. If you put a little fluorescent tag on those, you can actually tag each of the individual chromosomes relative to each other. And this is just a different way of looking at it. It's called the rainbow stain. Here again, we've got a male example here. This is what your karyotype is going to look like. Really good karyotype. I don't know if anybody's done any of these things. But all the chromosomes separated from each other, they're all mixed up together. If you've got your banding pattern like we had on that last slide, you can try and match up the bands and try and identify which of the chromosomes are which. Or here, again, if you've used these different fluorescent dyes, which are just different sequences in each of the chromosomes, you can figure out that this chromosome right here is one of the chromosome 1s. Oh, excuse me. This one over here is one of your chromosome 1s. And there's the other one. Here's chromosome 13. There's the other copy of chromosome 13. And then you can all line them up with each other. Why is this useful? Well, it turns out that in many cases, oops, oh, the images are not showing up here. That's strange. Okay, you can get chromosome translocations, and that's how you can end up with the color of one chromosome on one of these copies, which has been switched with one of these over here. There's an example in the textbook, which, again, is on my slides here, but didn't show up in the PowerPoint. And that is one of the things that happens in a lot of cancer cases. And so particularly the case that I was going to show, and it's on this slide, is what's called the Philadelphia chromosome. And it's something that people have known about for quite a long time. It causes a particular gene fusion, which leads to chronic myelogenous leukemia, which there's now a really good drug to treat. Anyone know what it is? Gleevec, Dr. Drucker, up the hill. Actually, now his office is down the hill at the South Waterfront. Developed. And so just by looking at the chromosomes and a karyotype, you can actually know what drug to use to treat your patient with.
method standpoint, you're tagging the chromosomes for certain patterns. That's how you, is that how you're labeling them? So here's the pattern. This is going to be green. If it repeats, the other one will be green as well.
Okay, so the question here is, how do you get these different colors? Basically, just yeah. I'm sorry, I'm paraphrasing your question here.
Like,
yeah.
This looks so simplified.
Right, and so basically, to be able to do something like this, you need to know a lot about the human genome and literally all of the sequences that are present in any one of the individual chromosomes. And so you have to have different sequences which are present in one of each of these 22 autosomes and in the sex chromosomes. And so each of those sequences, unfortunately, you can have little short pieces of those sequences. You will then put a fluorescent tag on. And so now you've got a collection of, we'll oversimplify here and say 22 plus 2, different sets of usually pretty short pieces of DNA which are specific to each of those individual chromosomes. And then what you do is denature them and then renature them with these little pieces that have those tags on them. And so that's the way you get that. Yeah, was the question over here?
Do you have to like do it all over the chromosomes? I noticed they're fairly evenly colored. Do you have to like, or do you just can you tag it once? Or?
Right. So the question is, they're really nicely evenly stained, and you would think if there's just one piece, it would only show up in one part of the chromosome. You're exactly right. So when I said 22, that would actually just give you little spots on each of the individual 22 chromosomes. So these are really actually mixtures of lots of different sequences which are specific to each of those individual chromosomes. And what that ends up meaning is that doing something like this is really beautiful, and you can put it in a textbook and you make a neat publication. It's very specific, at least in terms of looking at karyotypes, but it's really expensive. And so still, even now, mostly people do much more straightforward staining. And hopefully, we'll be able to see that here. So this is, in fact, a real human karyotype from the OHSU cytogenetics lab, got from a couple of years ago. This is what people actually really need to look at and try and line these things up. They're way better than me at trying to figure out how to actually get this to work. So you take that picture, look here, beautiful karyotype of all of those chromosomes. So going from something like this to something like that is, again, is to me is pretty amazing. So these are real data. And of course, that's interesting because you can now look at these cancer karyotypes. And unfortunately, this is a little hard to see here. But again, down at the bottom, these are what kinds of karyotypes they got. But this is a really extreme case. Many cancer cells have really messed up, and messed up is a nice word, kinds of karyotypes. So this particular one has 5 copies of chromosome 1, just 1 of chromosome 6, absolutely none of chromosome 17, just messed up. And there's arguments about whether this being really messed up is a consequence or a cause of the cancer. They're probably some of both, actually. Here's another example of one of those, completely missing chromosome 14, just 1 of 19, etc. So these are just really, really crazy, wacky kinds of karyotypes. Yes? Just to the cancerous cells, exactly. And so if you're talking about, biopsies, unfortunately, one of my colleagues' wives is probably having to have a biopsy in the near future. It's one of the things they're going to look at. Although what we're moving towards now, actually moving away from this, is just sequencing the whole darn thing, because it's getting relatively cheap to do that. Yeah, in the back? Yeah, so why do the 2 karyotypes look so different? We've got, oh, sorry, that's, okay, yes, I pushed the wrong button. If you go back to these, these are much nicer looking than the ones that I just showed. That apparently just has to do with the prep. And these things are apparently not completely straightforward to do. So, yeah, these are much more blobby. These are nice, long, and elongated. These are just a couple of examples. So one of the students actually was in this class, then went and got a job up in cytogenetics and then gave me some of these, which was really, really nice of them. Okay, other questions on, oh yeah, sorry, karyotype? Of the karyotypes? Yeah, so, okay, unfortunately I didn't have that image of the Philadelphia chromosome where you have these translocations that take place. And so it's not just that you've got different numbers of different chromosomes in some cases, but in some cases some of those chromosomes have moved around. And in the case of Gleevec and CML and the Philadelphia chromosome, if you see the Philadelphia chromosome, you know that's a chronic myelogenous leukemia. Turns out that the translocation happens in one particular place, ends up giving you a fusion protein in this kinase that Gleevec treats perfectly. So just by looking at a karyotype, you can design a therapy. Now, unfortunately, that's the exception rather than the rule. But that's certainly one of the things. And in fact, karyotypes are really pretty specific for lots of different cancers as well. It depends, again, depends completely on the cancer. And that's kind of beyond this course. But it's certainly one of the things that's really interesting. But as just a quick an aside there, yes, we're moving away from karyotyping now and just to whole genome sequencing, which is computationally a lot harder, but gives you a lot more information because it's literally giving you the sequence of all of these things. So I'm going to continue with this here. Let's see, somehow I've lost a bunch of slides here. But we'll just talk briefly about what's in any particular chromosome. So with those karyotypes, again, they're kind of blobs. What do we now know more about? So it's just an example here of chromosome 22, which is, it turns out, it's actually one of the smaller chromosomes that we have. And in fact, it was the first chromosome, human chromosome, to have the complete sequence done of. If you look at this particular chromosome, it's got about 50 million base pairs in it. That's pretty standard, again, for some of these smaller chromosomes. About 10% of that, it's got about 40 genes in it. Those 4 genes, it turns out, are mostly non-coding sequence. So the way that this is color-coded is basically each of these blocks here represents a gene, and anything which is gray is just extra stuff. We don't even really know what it is. And then even in the gene itself, here is pretty typical. It's about 30,000 nucleotides in length. Most of it is non-coding. So all these gray things here are not going to be coding for protein sequences. Each of the red ones are the so-called exons, which are actually going to give you your final protein at the end. This orange region over here gives you the regulatory information. Unfortunately, this is really variable. The orange region is often as much a guess as it is anything else. And it's quite possible that some of your regulatory DNA sequences are in these gray regions as well. Once you have this particular gene, the regulatory DNA sequences say time to make an RNA copy of this. That then gets translated into protein, and then you finally have the folded protein that actually does its job here. Fun DNA facts: if one base pair was 1 millimeter long, it would stretch all the way across Africa, just one genome. So it also gives you sort of the idea of the complexity and the absolute, yes, you can sequence whole genomes of cancer cells, but the amount of information there is so massive that picking and choosing and finding what's in there is really, really challenging. So just to finish up here, again, fun genome facts. This is the case now for the human genome. The haploid genome, i.e. just the copy you get from one of your parents, is about 3 billion nucleotides in length. Of that, there are about 21,000 proteins which are encoded, at least protein-coding genes. So we'll talk about later with splicing. Some of these can generate up to 30,000 different proteins. So yes, those protein-coding genes are relatively few numbers of them, but you can get lots and lots of different proteins actually out of them. Mean size, again, as I mentioned before, is about 30,000 nucleotides. Smallest number of exons, again, these pieces of protein-coding sequences, are 1. Largest is not such a big deal. More important is about 10 exons per gene. So those exons are going to provide you with the raw material to be able to do differential splicing. Which exon do you use? Which exon don't you use in terms of any one of those genes? So 10, and the combination of 10 different possibilities gives you lots and lots of possibilities for any given gene. And then about 145 nucleotides is the length of each of those any given exons. And hopefully you all remember that genetic code is a triplet code, and so that means you've got about 100 amino acids per exon. And it should not be terribly surprising that most domains are also about 100 amino acids. So if you think about mixing and matching domain, that domain has a particular structure. That structure gives you a function. Yes, thank you. By the end of the term, you'll all have it down. Well, you'll have it down. So that is very often what's happening, is you're moving and switching domains around, as we mentioned last time. So again, largest, smallest is not such a big deal. Probably most important, and this is why this is from this version of the textbook, this table, not a previous version, is the number of non-coding RNA genes. So we always used to think, RNA is important for the ribosome, tRNAs, and messenger RNAs. Well, it turns out there's a whole bunch of stuff in the human genome which are the non-coding RNAs. So these are RNAs which don't seem to have an obvious function, at least not in terms of translation or as a messenger RNA, but are clearly important. And we'll get back when we talk more about the genome in terms of why we think, or in fact we know in some cases, that they're really important. This number keeps going up. The number of protein coding genes is actually anything going down, but the number of RNA coding genes is going up. Pseudogenes, this is just copies of genes that are clearly non-functional anymore. Tells you a little bit about evolutionary processes. And then protein coding, 1.5% of our genome is protein coding, so it's a really, really small amount. And then about 50% of our genome is in repeated sequences. And we'll talk about what those are a little bit more later on. Yeah, 2 quick questions and we're done.
Does non-coding RNA gene, does that mean a piece of the RNA that doesn't get translated, transcribed?
OK, yeah, so the question is, what do I mean by non-coding RNA? Again, just paraphrasing your question. Yes, so it's an RNA which is transcribed, i.e. it's made from DNA into RNA, but it doesn't get translated into protein, or it doesn't get used for the translation process. Yeah, in the back?
How does the gene know what is intron, what is exon, and does it ever fall that the lesions get bigger?
Yeah, how does the cell know what's an intron and what's an exon? And much more actually problematic, how do we know what's an intron and an exon if we want to actually try and use this sequence? So we'll talk much more about that when we talk about splicing. See you on Wednesday.